anatomy and physiology. We're going to kind of come back here and start working through the last little section of this, which is that specific immune defense. So those third line defenses. So when we're looking at these ones, what they're really trying to do is find these foreign antigens. And these are going to be, really when we start getting the immune system recognizing, these are going to be little foreign molecular groups that they're going to recognize on these particular foreign molecules. It should be the foreign antigens that trigger an immune response and not self-antigens. And again, usually the body is able to tell these things apart. So what are some of these antigens? Well, an antigen is going to be just any molecule, technically, that the immune system is going to recognize as foreign when we talk about an antigen. What you can kind of see, and this is kind of showing this idea, let's say this whole thing right here is the antigen over here. You can see that each of these antibodies on this one here is binding to something that looks slightly different. The molecular group that the antibody or the T or B cell actually recognizes as foreign, the actual molecular group is referred to as an epitope. There can be, as is shown on this one, you can have multiple epitopes on a single antigen. A lot of times if you're like for the COVID, for example, if your body was making antibodies against it, it might make antibodies against multiple epitopes. If you get a monoclonal antibody injected into you, that might be something that was built towards a single epitope. So even with the foreign antigen coming into the body, a lot of times you're going to have multiple, let's say, antibodies or multiple T cells, multiple B cells that could recognize this particular thing as foreign based on the number of epitopes it might have on that one antigen. Immunogenicity is how much that antigen is actually going to trigger an immune response. The more immunogenic something is, the more robust the immune response is going to be against it. It has to do with size, what's exposed on it, how foreign the body recognizes it to be. All these things might increase or decrease the immunogenicity of a particular antigen. Haptens are something that are usually too small to serve as an antigen alone. These are very small molecules, so kind of like the toxin and poison ivy. But when it combines with certain body proteins, then the immune system can recognize it. And that's what happens on some of these hypersensitivity reactions, things like pollen, certain other drugs, poison ivy toxin. Those things normally in and of themselves couldn't trigger an immune response or that allergic response. But when it binds to a body protein, the immune system can then recognize it and respond to it. And in some cases does it in a way that we don't particularly like. And again, this is just showing you different things that can be antigens. So what we're usually worried about is some type of microbial virus, microbe, a virus, some type of worm, things like that. The body's also a lot of times going to try to reject anything that is a non-human cell, even if it was put into us on purpose, like a pig heart valve or a heart transplant, for example. The immune system a lot of times would still recognize that as foreign and is going to try to respond and attack it. And again, the thing I'm showing you down here is this whole molecule right here might be an antigen. Each of the different things on it might be an epitope. So again, single antigen might have multiple epitopes or antigenic determinants that the immune system is going to recognize and respond to. So what we're going to talk about here is just kind of a little bit of an overview of some of these things here, and then we're going to work through some of these different responses that we'll see in response to these different parts of the specific immune system. The specific immune system, it is not a immediate thing. It's not like the innate that's already there. This is going to take some time for the body to ramp up a little bit. Like it says there, that first time you respond to an antigen, it's referred to as the primary response. And a lot of the stuff we're going to talk about in these later shows, it takes a little while for it to ramp up. So it might take a week, 5-7 days, something like that. What we're going to see is the specific immunity, though, you do have the ability to set aside memory cells. And because of that, every exposure after that first one, we talk about it being a secondary response. And one of the things we're going to see about that secondary response is it's more robust. It tends to happen much quicker. And a lot of times it means you don't even actually get sick when you're exposed to that antigen a second time. We can look at the adaptive immune system or the specific immune system. We can subdivide it into two main parts. You have what's called humoral immunity, and this is using B cells and antibodies to go about attacking this stuff. Where cellular immunity, this is using your T cells, and these are T cells binding to and attacking cells that are inside a foreign body that's inside of a cell. So intracellular pathogens versus things like bacteria being in the bloodstream with humoral immunity. The development starts taking place early on in development, embryonic development even. Parts of that embryo, like it's saying there, are going to start developing these cells. Sooner or later, we're going to see it ends up in the bone marrow once you're born and things are kind of going along here. And again, it starts out from those hemocytoblasts. They go down the differentiation process. We're going to be worried more about the B and T lymphocytes in this one here. And again, that maturation process for each of these is going to be a little bit different. Both B and T lymphocytes have particular receptors on their surface that are going to have some type of specificity. There's quite a large number of these per cell. Each cell is going to have a particular specificity towards some foreign type of antigen. And you're going to have a large number of these cells that are present within the body. T cells have T cell receptors. B cells have B cell receptors on them. And what we're going to see is the B lymphocytes, they actually bind directly to the antigen in process there, and then that's going to go through a kind of a little bit further differentiation process, and we're going to see that it's going to do a certain thing where T lymphocytes have to have some type of antigen-presenting molecule facilitate this process. So they are going to have some type of antigen-presenting cell, or a helper T cell, for example, is going to have to process this stuff first and allow that T cell to become active against it. On T cells, these receptors that are going to deal with these foreign antigens are called CD receptors or CD molecules. And particular lymphocytes have certain subclasses of these, and it's part of the way that we differentiate them from one another. We're going to see a little bit later on, we'll talk about these CD4 and CD8 cells, which actually end up being helper and cytotoxic T cells, respectively. And that's what we're going to have to see right here. There is more than these two types of T cells, but these are the two main types that I focus on. Helper T cells, these are going to be ones that help activate B lymphocytes, as well as other immune cells, and cytotoxic T cells. They have these CD4 receptors in the plasma membranes, and a lot of times are referred to as CD4 cells, where cytotoxic T cells, they're going to release molecules like perforins that can help kill off cells that are infected with something. They have these CD8 receptors, and a lot of times they're referred to as CD8 cells. So if you ever see something that says a CD4 or CD8 cell, that's what it is actually referring to. T cell maturation, like I said, is going to happen differently than B cell maturation. It takes place in the thymus, so a lot of times we could talk about thymus-derived lymphocytes or T cells. Both T cells and B cells are going to start out in the bone marrow, but the T lymphocytes, when they're released into circulation, migrate to the thymus and are going to mature in the thymus. T cells act directly against infected cells here. Again, we could get into it, but I don't go that level of depth. Immunology is probably the course you want to take if you want to go in super deep. But T cells are going to release certain cytokines that are going to influence other cells. The other thing is when a T cell came in contact with the antigen or the epitope that is specific to, it can generate these long-lasting memory cells, which again help generate that secondary response and allow the second time you see an antigen for the body to respond much, much quicker. And it shows the process right here. They start out in the bone marrow, mature in the thymus, become educated, as they nickname it here. Where the B cells, they're going to be a little bit different. They start out in the bone marrow, finish their process in the bone marrow, before they're going to go to a lot of these secondary lymphatic structures, things like lymph nodes, that malt, the tonsils, things like that. This is where a lot of these lymphocytes end up when everything is said and done. Again, it's these thymusins that are in the thymus that are going to allow these cells to mature. Once they mature, again, they're going to travel out to these lymphatic organs and other sites within the body. The two main types that we're going to worry about, like I said, are the T helper cells that trigger the cytotoxic T cells, as well as the B cells. I always kind of say the T helper cells are kind of like the quarterback. They're in the middle of everything, kind of being as the general of the immune system, doing a lot of the command and control, coordinating the two branches of this specific immunity, where the cytotoxic T cells are more like infantry. They are doing cell-to-cell attacks here against foreign antigens, secreting, like I said, perforins that are going to lyse these infected or foreign cells. As you can see, there is a couple other types. I'm not worried about these ones for everybody here, but there are multiple types of T cells outside of the two that we focus on.